Welcome to Champion Medic. This is AQA Biology Unit 1, January 2011.
So question number 1 is simple knowledge recall, where they're asking us to label A. So we know from looking at the diagram that's the cell membrane. Just for the completeness of this whole paper, we're going to label other parts of the diagram of the prokaryotic cell. So you can see the flagellum, which is the tail coming out of the prokaryote. You can also see the non-linear DNA, which is the red outline there. You can see plasmid DNA, which are the small loops in red. And you can also see the cytoplasm, which is the other bit, and the
70S ribosomes.
70S ribosomes, yep.
There's also the slime capsule on the outside.
Yeah, so we're finished there. So part B, what two structures are present in an epithelial cell? So an epithelial cell is a eukaryotic cell, but those two structures aren't present in the prokaryotic cell above. So we know common one is nucleus. Another answer is mitochondria, Golgi, ATS ribosomes. Because as we've just gone through in prokaryotic cells, we've got 70S ribosomes. Nucleus, and the other one is non-linear DNA and plasmid DNA. So multiple answers that you can put down for this question.
If you refer to question 1 on the last video, you'll see the full diagram of the eukaryotic cell as well.
And another thing that you can notice is this question is the direct opposite of the last paper, where they wanted two features of a prokaryotic cell, but it's not present in a eukaryotic cell. So it's important that you realize that you need to learn this kind of knowledge because it keeps on coming up in the early knowledge questions. So part C is with regards to microscopes, and they want you to compare the TEM, transmission electron microscope, and scanning electron microscope, SEM. So a main advantage of the TEM compared to the SEM is that it has a higher magnification and resolution. That's a key advantage, and as a result of that, you can see internal structures much more clearly in the internal structures of the cells. Another so following the exam paper, the main advantage of the scanning electron microscope compared to the TEM is that you can obtain 3D images. Right, so do you want to go through other advantages and disadvantages of the microscopes?
So the limitations for each one of them, the disadvantages. For the TEM, or well, for both of them, the sample must be dead because they have to be placed in a vacuum. So if you get like a long 6 marker, you want to go through some of these points. And especially for the transmission electron microscope, so it'll be difficult to prepare samples. and you'll get the presence of artifacts, which are, they're just, when you view the image at the end, it's just like a blur or something that isn't present in the actual sample, but it occurs in the image due to, like, because it's difficult to prepare, you'll get like a blur or something in the image, and that's known as an artifact.
So, for example, dust particles that may get into the sample during the preparation process. Another disadvantage of the transmission electron microscope is that it can only be used on thin specimens. So during the preparation process, you have to make sure that the sample is quite thin, so that the beam of electrons can be transmitted through the specimen. So disadvantages of the scanning electron microscope is that it has much lower resolution images. And another advantage of the scanning electron microscope is that you can use it on thick specimens, as in you don't have to make sure that the specimens or the sections have to be thinly cut.
Okay.
So the next question.
Part D. This question asks to find the actual width between points B and C. So if you measure it, you should get 2.1 centimeters. And if you convert that into millimeters, you get 21 millimeters, and then convert it into micrometers, 21,000 micrometers. Then use the equation magnification equals image size over actual size, and just replace all of your information into there. So the question says 50,000 is the magnification. The image size is 21,000, and the actual is what we're trying to work out. Rearrange, you should get 0.42 micrometers.
Right, so part E is asking us to describe the graph and trying to obtain work out what the relationship between the two factors are. So if we look at the graph, we can see that as the height above sea level, which is on the x-axis, increases, we see a decrease in the number of deaths per 10,000 from cholera. So that's going to be your first mark point, and that's negative correlation. You can also say that to get you the mark, just from interpreting the graph. And if you look, you can see a much more shallower decrease, I think. If you look at point 24 to 26 meters on the graph, that's where you see, around 24 to 26 meters is where you see a much more shallower decrease. So that gets you your second mark. So mentioning a figure from the graph gets you your second mark.
So if you say decrease at 15, for example, you'll get a mark.
Yeah, it gives you a range between 14 to 30 meters, so any number between there. The second question is asking us to describe the induced fit model. So this is a concept in enzymes where the active site of the enzyme is not complementary to the substrate, which means that the active site of the enzyme has to kind of mold around the substrate. So it's kind of flexible in that aspect. So it changes the shape of the active site, and that way it can form an enzyme substrate complex. So if you look at the diagram, In the lock and key model, which is the model we're taught at GCSE, you can see that the active site is directly complementary to the substrate, so it can bind in straight away. Whereas in the induced fit, the substrate is not exactly the same complementary to the active site of the enzyme. Therefore, the active site has to kind of change its shape, kind of mold around the substrate, and therefore then on bind to form the enzyme substrate complex. So that's the difference.
So to answer the questions of part 1, you would say the active site isn't complementary. And that it changes shape, so you could say it's flexible. And it allows an ES complex to form, sorry.
So part 2 is asking us describe one way in which the lock and key model is different from the induced fit model. So we've just gone through that. The reason is because the active site does not change shapes or it does not mold around the substrate, because in the lock and key model the active site is already directly complementary to the substrate. So question 2b is talking about the concept of competitive inhibitors in biology, and so it tells us that PABA is the substrate and sulfanilamide is a competitive inhibitor which has a similar shape or similar structure to PABA. So what essentially a competitive inhibitor is that it's a substance that has a similar structure or shape to the substrate, but it blocks off the active site, which prevents the real substrate from binding, which results in lower rate of reaction because you get less enzyme substrate complexes forming because less of the substrate is binding. So this question has come up very frequently in past papers. They just keep on changing the situation in which it's around the context of it. So I would strongly recommend that you learn this concept of competitive inhibitors in biology.
3A.
Biology, yeah. So 3A, we see a diagram of the heart, and part 1 is asking us which of the blood vessels, so from D to G, carries oxygenated blood to the heart. So oxygenated blood to the heart. So if blood is being carried to the heart, it's only carried in the veins. So if it's oxygenated blood, that means, and it's being carried to the heart, that means it must be coming from the lungs. Therefore, it's the pulmonary vein, and that's G in the diagram, because we know that the pulmonary vein is connected to the left atrium. Yeah. Then part 2 is asking us which blood vessel carries deoxygenated blood to the lungs. So if it's carrying blood away from the heart, that means it must be an artery. And if it's carrying deoxygenated blood, we know that that's the pulmonary artery, and that's E. Right. Just for completeness, to label the rest of the um, blood vessels, D is the vena cava. The reason why you see 2 entries into the right atrium is because you've got the superior vena cava and you've also got the inferior vena cava, but you don't really need to know um, like what the difference is between the two. But it's nice to know just that there are 2, so you don't get confused by the diagrams. And F is the aorta. and so they alters the artery the oxygenated in which the oxygenated blood leaves the heart to go to the rest of the body. So question 3b, why do the semilunar valves open? Give an explanation. Well, for the semilunar valves to open, the pressure in the ventricles need to be greater than those in the artery. So the pressure below the valve has to be much stronger or greater. Make sure you use the word greater, not just great. So it has to have the ER because you need that comparison factor between the ventricles and the arteries. Yeah. So question 3c. What is the importance of this time delay that's created by the AVN? And the idea is that it allows the atrium to empty its blood, and whilst that's happening, the ventricles fill up with blood, and that's to ensure that process takes place before ventricular contraction, because you want to ensure that the ventricles are contracting when they have blood filled in them. That's the idea.
If there wasn't a time delay, then blood would rush back into the atria when the ventricles contract, and that's we just don't want blood to go backwards in the vessel.
The next question is a maths calculation question. Do you want to go through it?
Yeah, so it says calculate the athlete's stroke volume after training. So the values for after training, we know the cardiac output is 5,000 and the heart rate is 55. So we just use this equation and rearrange to get stroke volume equals cardiac output over heart rate, which gives you a value of 91, I believe.
Yeah, 90.9 or 91.
We'll just put 90.9.
One thing that I just want to say is make sure that you guys learn the equation in the purple box off by heart, because I don't think the exam gives it to you, does it?
No, it doesn't.
So it's a simple equation, but you don't want to lose 2 marks on a relatively easy question. So I'd recommend that you guys learn it.
Part 2. The question tells us that the cardiac output is the same and the stroke volume increases. So we can use our own logic to realize that the heart is becoming more efficient, but we need a more scientific way of saying that. So we can just say sort of contraction or an increase in size of the ventricles. Either of those would get you the mark. Question 4.
So, question 4a is a simple question, knowledge recall, that's asking us what is an unsaturated fatty acid. So we know that unsaturated fatty acids contain a carbon-carbon double bond. And this question is 2 marks just for writing carbon-carbon double bond. So yeah, it's very generous, I think, but you don't want to be losing marks on this question because it's too easy for it to lose marks. The next question, 4b, is asking us to look at the graph. So you can see we've drawn a similar graph to the one that you can see on your paper. And it's asking us, do the data show that eating omega-3 fatty acids prevents coronary heart disease? So the first thing you want to do is give a description of the relationship between the amount of omega-3 fatty acids eaten compared to the risk of coronary heart disease. So whilst it's not given exactly in the mark scheme, you can say something along the lines of this: as the amount of omega-3 fatty acids eaten per day increases, the risk of coronary heart disease increases. So that's the negative correlation factor. Also, it's key to mention that correlation does not mean causation. Um, because the through the negative correlation shown, that doesn't mean that's necessarily the case, because it can be attributed to another factor. So you can also give an example if you think of one. So the correlation does not mean causation answer, and maybe due to another factor, comes up quite frequently in mark schemes for these evaluation questions. So I'd recommend learning what those two phrases mean really well, and then implementing them into answers where it's appropriate. Question 4C. Part 1 is asking us to name bond X. So we know that's the glycosidic bond. Can't even speak. That's because it's a carbohydrate. It's a sucrose molecule, and it's between a glucose molecule and a fructose molecule. So we can identify the glycosidic bond from there. Part 2 is asking us give one way in which triglyceride is different from cholesterol. So we know that a triglyceride contains 3 fatty acids and a glycerol molecule, whereas cholesterol doesn't. And we know that from our own knowledge, and then we compare that to the information given about cholesterol in the extract. The next question is asking us how many molecules of water would be produced when 1 molecule of cholesterol is formed. So this question is actually quite kind of a tricky question, though it's only 1 mark. And to work out the answer of 9 is 9 molecules of water, you need to look at the diagram that they've given you of elestra on your paper. And if you look at it, you can see that there's 8 R groups. And what these R groups are, it tells you in the extract, in the sentence above, the letter R shows where a fatty acid molecule is present. So for those fatty acid molecules to have been attached to this sucrose molecule, a glycosidic bond, I think, would have, a condensation reaction would have had to take place. So that's going to produce 1 molecule of water. So that gives us 8 molecules of water from the 8 R groups, which are the fatty acids attaching via condensation reaction. Your 9th molecule of water is coming from the glycosidic bond in the middle, which is from the attachment of the glucose molecule to the fructose molecule. So that's bond X. That's your 9th molecule of water. Hope that made sense. Yeah.
Question 5.
So question 5 is asking us to explain why the student dried the outside of of each of the potato cylinders, and the reason is because each of the cylinders we know have different masses, as in the amount of water present is different, and therefore the amount of water is going to affect the mass of the potato cylinder. So you're going to get an inaccurate representation of the results. So 5B.
5B is a calculation, and it's asking how you would convert from a 1 mol dm cubed solution to a 0.2 mol dm cubed solution and get 20 centimeters cubed of that. So we start off with 1 mol dm cubed of sodium chloride, and we want to get to 0.2 mol dm cubed. That's dividing by a factor of 5. And so if we have a total of 20 centimeters cubed of solution that we want, the amount of sodium chloride that will be in there will also be a factor of 5. So you would get 4 centimeters cubed of sodium chloride. That would leave you with 16 centimeters cubed of water. And that will be your answer.
So the next question, 5C, is asking us the advantage of calculating percentage change in mass rather than just normal change in mass. The reason is because you can make easier comparisons between the potato cylinders, because each of the potato cylinders have a different starting mass. Make sure that you're specific by using words such as different starting masses, not just different masses.
Part D.
Part D is asking us about the importance of repeats, and the reason is because you can calculate a more reliable mean, and the reason is you're able to first identify anomalies and remove those anomalies, and after that you can calculate a mean. So question 5E is asking us about the isotonic point. That's the concept in biology. So it's the place where the concentration of the sodium chloride solution, or in other cases sucrose solution, has the same water potential as the potato cylinders. So if you look at the graph which we've drawn, it's the point where the results, so your straight line, your line of best fit, sorry, crosses the x-axis. So that's the point where everything is equal. So in this case, it's 0.35 mol
This
dm
is because
cubed.
there's a 0 change in mass, which means the water
Yeah.
potential in the solution is the same as the water potential in the cylinders, so there will be no net movement of water, causing no change in mass. Question 6.
I recommend that you guys read the passage first, just because it will be much easier. So 6A, so just why the vaccine is offered to this specific age group, and the reason is because, is that they are unlikely to initially carry the HPV vaccine because of their age. If the girls are already infected, then the vaccine may not work because it doesn't provide that initial protection first. So 6B is asking us, explain why this vaccine doesn't protect against other types of viruses. And the reason is because of the idea of the antibody and the antigen being complementary in shape. So other types of HPV would have different antigens that have a different shape or different structure to this antigen. Therefore, you're going to be unable to create memory cells for the other types of viruses because they have different antigens, and because the antibodies won't be complementary to the antigens, they can't bind. So there will be no formation of an antibody and antigen complex. So question 6C, use your knowledge of immunity to suggest why 3 injections are given, because you can get more antigens. And if you have more antigens like present in the body via a vaccine, you're going to have more memory cells being produced for the secondary response, and as a result of that, you're going to have more antibodies being produced. So your secondary response is going to have a faster rate of production of antibodies, because you have more antigens and more memory cells being produced from the first primary response. Part D.
It will take a long time for us to know if the vaccine has worked because, firstly, it will take a long time for cancer to develop itself. So cancer takes many years to develop. And it also will take time for there to be enough sexual activity for them to even come into contact with the virus. So those two things combined means it will take many, many years for us to even see if the vaccine works. So question E.
So part E is asking us, why do women who have already been vaccinated still need to be given smear tests? And the reason is because cervical cancer can be caused by other types of HPV. I think that's the main reason, because this vaccine, as it says in the passage, only protects against one, has proved effective against HPV type 16 and 18, but we don't know if it can treat other types of HPV. So that's the reason why. Because once again, cervical cancer can be caused by other types of HPV, but this vaccine only protects most effectively against type 16 and 18, as it says in the passage. So question 6F, give a reason why it's important to vaccinate a large number of people to prevent the spread of HPV in the population. And the reason is to ensure that the virus cannot replicate and that it's fully destroyed, and therefore you haven't got any carriers within the population. And as a result of that, even if you have non-vaccinated people being in contact with vaccinated people, it doesn't really matter because the virus has then been destroyed. So this comes under the concept of herd immunity. I think that's the common phrase for it. But that just saying the word herd immunity doesn't really get you any marks. And make sure that you guys are like really specific. So saying virus is destroyed gets you a mark, but saying disease is destroyed doesn't get you the mark because it's not specific to this passage, which is just talking about viruses and not other diseases caused by bacteria or fungus, for example. So the next question, 7A, is wanting us to talk and describe about pulmonary tuberculosis. So this topic is not actually on the new specification. It's the old spec. So it can't come up as a knowledge question. However, it can come up as applied application question, kind of applied to this scenario. So just for the completeness of the paper, we're going to go through it briefly. But you don't need to learn through it in detail, just have a general idea of the whole process. So the first thing that we know is that TB is a airborne disease. Therefore, it's going to be transmitted through droplets in the air. So that's the first thing to note, that the bacteria is going to be transmitted in air droplets or droplets in the air. So after the TB has entered the body of a person, the bacteria are then going to be engulfed by phagocytes.
So this is the immune response.
And after the immune response has finished, the bacteria are going to become dormant, or they're not going to be active anymore, but they're still going to be present within the body.
Because they've been encased in a granuloma, for example.
Yeah. So after that, if if the body becomes immunosuppressed, then the bacteria will begin to replicate again. So the word immunosuppressed means that if the person's body becomes into a weak state of immune system, so if the immune system becomes weak, the bacteria are going to begin replicating again.
So if your body is combating another, like a virus or another bacteria, your immune system will be weakened, in which case the bacteria for tuberculosis will become activated.
So, for example, if you have cancer, you're going to be immunosuppressed because you have a weak immune system. Therefore, that's why you're more likely to get opportunistic infections. For example, in this case, TB. TB. Um, so after that, after the bacteria have begun to replicate, you're going to have the bacteria destroying alveoli, which are the sacs in the lungs where gas exchange takes place. So if your alveoli are destroyed, you're going to end up with fibrosis, which is the concept of scar tissue in the lungs, and the scarring means that your connective tissue is not going to be the same again. So you're going to have a much lower, so less surface area or lower surface area because of the damage caused. And if you have a lower surface area, you're going to also increase the diffusion pathway, which means it's going to take longer for gas exchange to take place, process such as gas exchange to take place. And that essentially means you're going to have the bacteria entering the blood, and that's just going to spread to other organs within your body. So then you're just going to get your whole body shutting down afterwards. So I think it's called multi-organ failure or something like that.
Yeah, it could be.
Yeah. Um, as we said, you don't need to know it in complete detail, but just have a general idea of what takes place, because it can come up in application questions. Question 7B. Why do people with emphysema feel weak and tired? Another 5-mark question. This question, I think you do need to know off by heart really well. And so emphysema is when the alveoli initially rupture or break down. It's really key that you use words that we've just said, such as rupture or breakdown. Do not say just damaged, because that's too vague for the mark scheme. So if your walls have been, if the alveoli is ruptured, you're going to have a lower surface area. And having a lower surface area is connected to an increase in the diffusion pathway, or a longer diffusion pathway. You're also going to have a lot of elastic tissue. So that's the stretchy tissue kind of thing that's involved around the alveoli.
It's called elastin.
Yeah, don't write stretchy material. Make sure you guys actually use the real word, which is elastin.
The alveoli can't recoil.
Yeah. So this question is just about making direct links.
So it's harder for you to expel the air that's already in your lungs. And that reduces the diffusion gradient.
Which means if you're going to have a lower diffusion gradient or reduced diffusion gradient, you're going to have less oxygen being able to enter the blood. That means less oxygen is taken to the heart muscle or cardiac muscle, and that means that the heart is unable to respire. Which means you can't produce ATP for certain processes. So if you have less respiration, that means less ATP energy is being produced, and that links directly back to the question why people with diseases such as emphysema feel weak and tired. That's the key link back to the whole thing. So we'd recommend that you guys learn this off by heart as well. Like really well with detail, just because this kind of topic of the alveoli comes up frequently in papers as knowledge questions. So you don't want to be losing marks on a relatively easy to understand topic. So if you don't understand this topic well, I strongly recommend that you guys just spend a couple of hours going through it really well to make sure that you're really clear on this topic.
That's the end of the paper. Thank you for watching. We'll be posting more videos soon.
Um, so if you guys like the video, make sure you put a like and subscribe and share. See you next video.